Okay, let's begin. So I want to go over this paper, and I'm just going to go through the abstract because I want to make a shorter video today. But here, prolactin-induced activation of nuclear factor kappa B in bovine mammary epithelial cells, their role in chronic mastitis, which is inflammation of the breast tissue. So the reason why I want to go over this is because I want to bring attention to the fact that prolactin is able to influence immunity, and particularly prolactin is able to increase the pro-inflammatory state when prolactin is in high concentrations. In fact, the prolactin receptor is very similar in structure to, it's a cytokine receptor. It basically functions similarly to other cytokine regulatory hormones, right? So here, prolactin-induced activation of nuclear factor kappa B, this is the method by which prolactin is increasing this causing this pro-inflammatory state in a lot of people. So remember, nuclear factor kappa B, I've done previous videos on it. I actually did a series of 4 videos going over a review regarding what nuclear factor kappa B is. Just know that it is a what's it called? A heterodimer. So there are multiple different things that could be nuclear factor B, but you basically have a heterodimer of 2 different things that come together, and then this will translocate into the nucleus of the cell, and it'll bind to DNA directly. And by binding to DNA, it can promote the activation of certain genes. And so it will activate these genes, and a lot of these genes tend to be pro-inflammatory genes. Some of them are will cause differentiation of certain cell types, especially the differentiation of T helper 1 and T helper 17 cells, which are very pro-inflammatory T cells. Regardless, prolactin will, like many other cytokines, interact with the nuclear factor kappa B pathway. It'll find some way to basically activate nuclear factor kappa B, which then results in the activation of these frequently pro-inflammatory cytokines. So in this particular case, I just wanted to highlight this prolactin-induced activation of nuclear factor kappa B in bovine mammary epithelial cells. This activation has a role in chronic mastitis, which is inflammation of the breast. So let's look this up real quick. So it's a prolonged inflammatory breast disease, and little is known about its cause. So let me go to Wikipedia, because Wikipedia usually has a good, yeah, okay, so let's keep this PG. Okay, causes. Since 1980s, mastitis has often been divided into non-infectious and infectious subgroups. However, recent research suggests that it may not be feasible to make divisions in this particular way. And it's been shown that the types and amounts of the potentially pathogenic bacteria in breast milk are not correlated to severity of symptoms. And moreover, although only 15% of women with mastitis in this study were given antibiotics, all recovered and few had recurring symptoms. So many healthy breastfeeding women wishing to donate breast milk have potentially pathogenic bacteria in their milk but have no symptoms of mastitis. So, so it can often be, or about 15% of women require antibiotic treatment, but so it can be brought on by bacterial infection or infection of some form. But here we're saying that in bovines, in cows, this can be caused due to excessive prolactin. So let's start. We sought to determine whether prolactin, also known as PRL, could influence the neutrophilic inflammation, and this is inflammation that is caused by neutrophils. You know what? Let's just look up what a neutrophil is explicitly. So it's a type of white blood cell, and they're the most abundant type of granulocytes, and the granulocytes are a category of white blood cells in the innate immune system, characterized by the presence of granules in their cytoplasm. So basically, they can release certain things into the environment. They have basically chemicals that are within the granules, and they can often release these granules into the environment to elicit different effects. So neutrophils are a type of the granulocytes, and they make up 40 to 70% of all white blood cells in humans. So this is when a lot of us think of white blood cells. This is one of the major white blood cells that we think of, and so they form an essential part of the innate immune system, with their functions varying in different animals. So they're formed from stem cells in the bone marrow, like many other cells. So with that, one thing that I will also talk about is that they migrate through the blood vessels and then through the interstitial tissue following chemical signals, which could include interleukin 8, C5a, which is a protein fragment released. Yeah, it's just a protein fragment. These other things, and hydrogen peroxide, and this process is called chemotaxis. And so with these chemicals in the environment, they tax, or they move towards the chemicals in order to fight an infection. So with this, and just so that we're all clear, here we have the multipotential hematopoietic stem cells, which I would presume would be in the bone marrow. And so this can multiply, but sometimes they can differentiate into these common myeloid progenitors or the common lymphoid progenitors. And so these lymphoid progenitors can turn into the natural killer cells, which are large granular lymphocytes, or they can turn into the small lymphocytes, where you can get the T lymphocytes and the B lymphocytes. And the B lymphocytes can also turn into the plasma cells as well. So this is one branch that they can go to, being a part of the lymphoid system. And then another one is where they can go down this myeloid path, where they can turn into red blood cells, or they can turn into mast cells, or myoblasts, or megakaryocytes. And the megakaryocytes would turn into platelets, if I'm remembering this correctly. And then the myoblasts can turn into these other types of things. And one of those things could be the basophils, neutrophils, which this is what we were talking about earlier, the eosinophils, or the monocytes. And the monocytes can turn into macrophages, which are referred to because they frequently will consume other things. So just keep in mind, this is sort of the basic, overly simplified structure of what types of white blood cells you have. Let's come back to this. So this could result, or could influence, the neutrophilic inflammation that characterizes chronic mastitis. So most of the genes encoding inflammatory proteins depend on the nuclear factor kappa B, also known as NF-kappa B, for their expression. So a lot of the pro-inflammatory proteins and or cytokines, these are the result of the nuclear factor kappa B activation of their genes. So we address the hypothesis that immunomodulatory activities of prolactin might arise from an increase in nuclear factor kappa B activity. So the MAPT cells, which are a bovine mammary epithelial cell line, these were stimulated with increasing concentrations of bovine prolactin. So level of nuclear factor kappa B binding activity was measured, and mRNA was evaluated for interleukin 1 beta, interleukin 6, interleukin 8, granulocyte macrophage colony stimulating factor, also known as GM-CSF, interferon gamma, and tumor necrosis factor alpha, and other cytokines known to require nuclear factor kappa B. And all of these, or a lot of these, are cytokines which are known to require nuclear factor kappa B for their maximal transcription. So this is playing, if you can see more of these cytokines, this could tell you, or I should say, they measured the mRNA, so they measured the transcription from the DNA into the mRNA. They are measuring this for these cytokines, which would indicate to them that nuclear factor kappa B is being activated if they are higher than normal. So prolactin activated nuclear factor kappa B, and maximal nuclear factor kappa B activation was weaker with prolactin than with tumor necrosis factor alpha at 30 or 180 minutes post-stimulation. So wait. Levels of nuclear factor... Okay, I might have to reread that later. So mRNA expression of interleukin 1 beta, interleukin 6, interleukin 8, GM-CSF, tumor necrosis factor alpha. Prolactin activated this And maximal nuclear factor kappa B activation was weaker with PRL than with tumor necrosis factor alpha at 30 or 180 minutes post-stimulation. Okay. In addition, prolactin significantly amplified in a dose-dependent manner mRNA expression of interleukin-1 beta, interleukin 6, interleukin 8, GM-CSF, and tumor necrosis factor alpha. So prolactin significantly increased it. So that's basically telling us that nuclear factor kappa B was definitely being stimulated. So we measured PRL concentrations in blood and milk from healthy and chronic mastitis-infected cows and studied the relationship between the prolactin concentrations and the degree of inflammation in the mammary glands. Mammary gland as indirectly assessed by somatic cell counts. And somatic cell counts, these are the body cells. So plasma prolactin did not differ significantly between the healthy and chronic mastitis-affected cows. Interesting. Milk prolactin concentration was significantly increased in chronic mastitis-affected porters with the highest somatal cell count and had positive significant correlation between SAC and as well as between the number of neutrophils present in milk samples. So I would presume that the somatic cell counts would be measuring the white blood cells that were present in the milk, and they're saying that milk prolactin concentrations was significantly increased in the sick cows, and they also had a lot of neutrophils in the milk of those that are suffering from this chronic mastitis. So as well as the number of neutrophils present in the milk sample. So maybe this somatic cell count is not regarding the neutrophils. I wouldn't be able to tell you exactly what this is doing. Anyways, the present findings showed that prolactin promotes an inflammatory response in bovine mammary epithelial cells through nuclear factor kappa B activation and suggests a role in prolactin in pathogenesis of chronic mastitis. Now, what I'll have to go back to is looking to see if prolactin can be produced at the location. So prolactin Wikipedia, and let's see if prolactin is secreted from the pituitary gland. So it appears as though it's only coming from one location predominantly. So going back to this, it appears as though the bovines or the cows did not have elevated prolactin in their blood serum, but they did have elevated prolactin in the inflamed tissue. Is that, at least that's what it appears to be. So yeah, so they say that the plasma PRL did not differ significantly between the healthy and the chronic mastitis affected cows. So yeah, there's not much different here. However, the milk prolactin did have a significant increase. So that is interesting that there is this localization of the prolactin into the milk. I mean, I guess it makes sense, but yeah, I'll need to mull over that because I would have presumed that. I mean, it could be possible that prolactin frequently will come out of the serum. So even if you elevate prolactin in people's bodies, it will quickly try to deposit itself or at least get out of the blood very quickly. So you wouldn't see that much of a difference in the blood plasma. So that would be, I should look that up a little bit because perhaps even if you do have elevated prolactin levels, perhaps it will very quickly deposit itself in some way. So yeah, I think, and granted, another thing that we need to consider is prolactin in of itself can be upregulated by inflammation, right? So it could have been, this doesn't tell you whether or not prolactin was elevated and therefore caused this mastitis. It could have been that somewhere along that metabolic pathway, something got triggered, and then it started this positive feedback loop of a bunch of different chemicals that are, or metabolic pathways, if you will, that are basically cycling cell signaling pathways that are going over and over again. And if prolactin is just one of those chemicals that gets upregulated, yes, you could say, oh yeah, prolactin is elevated and then it's causing inflammation. But the other thing that you need to consider is it might be something that is upstream or downstream of prolactin in that cycle that could also be causing the elevated prolactin levels, which also result in, which that cycle also results in more inflammation. So, it's very hard to tease apart all these variables. Just know that if you activate any part of that cycle, you could theoretically create a self-fulfilling cycle that maintains homeostasis at that particular state. So, continuing, the present findings show that PRL promotes an inflammatory response in bovine mammary epithelial cells through NF-kappa B activation and suggests a role in prolactin in the pathogenesis of chronic mastitis. So, with that, I think we're going to head on out, but just one thing that I want to consider, or I want to bring up, is that there was some genetic sequencing, or I should say amino acid sequences of the SARS-CoV-2 spike protein that definitely stood out to me, in that I thought that there might have been a strong link between if your body were to generate an antibody against this particular part of the spike of the SARS-CoV-2 coronavirus, that antibody would be an autoantibody, and it would actually attack your own peptide hormones, and it would specifically attack a peptide that would end up causing the release of prolactin, or cause an elevated, cause the expression of more prolactin. So this is what I'm worried about, is that this autoimmune disease that would be the result of creating an antibody of the wrong type, that it would cause elevated prolactin levels, which would cause more inflammation down the road. And through the mechanism would be through nuclear factor kappa B. And granted, this would be some of the reasons why cortisol and some steroid treatments such as dexamethasone, this might explain possibly why it has some, what would the word be, some anti-inflammatory benefits, and why it did show to be beneficial in, it did prove to be beneficial in some serious forms of SARS-CoV-2 at reducing the inflammation, which allowed the body to heal. So, with this, I think I'm going to head on out, but if you have any questions, please leave them down in the description, and hopefully I will get another video out soon. So, thank you.